Hello to all those who are watching this video. This is the video for the students of class 11th biology. Okay, so Nivedita is here by taking a detail of the topic protein, which is a biomacromolecule. Okay, so in this video we will study about the proteins, its structure, its components, okay, functions and types as well. So begin with what they are. So proteins are polypeptide chains and made up of amino acids. So there are 20 amino acids known, okay, and which join together by polypeptide bond between amino acid and carboxylic group. Okay, means CONH. This is, and these are the most important and abundant intracellular organic biomolecules. They are just means all amino acids, they are linked by the peptide bonds that I told you, that is CONH. Okay, and amino acids, we have the example of alanine, valine, arginine, leucine, histidine, etc. So proteins are considered as heteropolymer, and these amino acids are divided into two main types on the basis of their utility, that is essential amino acids and non-essential amino acids. Now it is clear that amino acids are the basic unit of proteins. Means जब बहुत सारे amino acid जुड़ेंगे तो वो एक polypeptide chain बनाएंगे. Okay, and chains will constitute the protein molecule. Okay, so there are two kinds of amino acid. Essential amino acids, इसको हम ऐसे याद रख सकते हैं, it is very essential to take these amino acids in our food. मतलब ये amino acids हमारे खाने में होना चाहिए. And non essential means they are not required. Because why? Because our body can prepare these amino acids, तो हमें ज़रूरत नहीं है उसको food में लेने की. Okay, so these are the structure formula of amino acids: glycine, alanine and serine. If we talk of the functions, the main function of protein in the living cell are transport of nutrient across the membrane. They produce enzyme and proteins. Just keep in mind, collagen is the most abundant protein in the animal world. Okay, in the animal world. And it helps in fighting with infectious organisms. These are helpful in movement of muscles. For example, myosin and actin. Okay, they help in maintenance of pH and regulation of the volume of body fluids. They help at the time of injury in blood clotting and act as antibodies and provide immunity. They help in growth and repair of body tissues. And some proteins functions as hormones and some function as enzymes and catalyze the reactions. Okay. Denaturation. What is denaturation of proteins? When proteins are exposed to extreme change in pH, acids or temperature. Okay. The weak bonds holding the tertiary and quaternary structure gets disrupted. So the protein unfolds, and this unfolding is known as denaturation of protein. Okay, and that time protein may lose its function. Okay, so denaturation is not strong enough to break peptide bond, thus primary structure remains unaffected. A denatured protein may spontaneously refold into its original structure when suitable conditions are re-provided. So this is called renaturation. So we have this four types of proteins. Primary structure means on the basis of their structure. Primary structure, secondary structure, tertiary structure and quaternary structure. So primary structure, it is a basic structure of protein in which a number of polypeptides are involved having sequence of amino acids. Okay, the first amino acids of sequence is called N-terminal, जहाँ पहला होता है, And the last amino acid of the peptide chain is called C terminal. ओके, पहला वाला N और लास्ट वाला C. तो इसमें प्राइमरी स्ट्रक्चर में जो अमीनो एसिड्स होते हैं वो एक लीनियर फैशन में होते हैं. Secondary structure of protein, it forms threads. Okay, the threads form the helix. So there are three types of secondary structure: alpha helix, beta pleated, and collagen. In alpha helix, the polypeptide chain is coiled spirally in right-handed manner. You will get the diagram in the next slide. In beta plated secondary proteins, two or more polypeptide chains are interconnected by hydrogen bonds. And in collagen, there are three strands of polypeptides coiled around one another by hydrogen bond. Okay. And this is the secondary structure of protein. Okay, उसमें अल्फा हेलिक्स मिलेगा, बीटा प्लीटेड मिलेगा और कोलेजन मिलेगा. And the tertiary structure, these are long protein chains folded upon itself like a hollow woolen ball. Okay, again उसका जो वूल का बॉल बना होता है उसके जैसा होता है, to give the three-dimensional view of the protein. There is bending and folding of various types to form a hollow woolen ball-like spheres, rods or fibers. कुछ भी हो सकता है. Tertiary structure is stabilized. This is very important to know by several types of bonds: hydrogen bonds, ionic bonds, okay, van der Waals interaction, covalent bonds, and hydrophobic bonds. So many bonds are involved. It gives information about a three-dimensional conformation of the protein. Okay, for example, myoglobin. So tertiary structure is helpful for many biological activities of proteins. There is a differences between the alpha helix structure and beta plated structure. Okay, you can see here the diagram A and B. And if we talk of, okay, this is secondary and the diagram is showing the secondary and the tertiary structure of the protein. Okay, and in quaternary structure, each polypeptide develop its own tertiary structure and functions as subunit of protein. For example, hemoglobin. In adult human hemoglobin, 4 subunits are involved. Okay, in two subunits are of alpha type and two subunits of beta types. Okay, so this is quaternary structure. Certain proteins consist of an assembly of more than one polypeptide or subunits. Thus the individual polypeptide or subunits are arranged with respect to one another. Okay, so already I told you कि इस तरीके के जो हुआ करते हैं उसके example we have the hemoglobin, lactic acid, dehydrogenase enzyme, these all. So in this type of structure, this is seen in the oligomeric proteins, that is proteins having two or more polypeptide chains. So thank you so much.